Joseph, you look different without your mask.
Yes, I have a little bit more facial hair.
Are you letting it go?
Yes, at least for now.
Okay.
Who needs showering? I have the chlorine from the pool.
All right. Whoa. Why is that doing that? I'm having some technical difficulties with my computer. Can y'all see the basic statistics thing? Yeah, okay, thanks, Joseph. Okay, Joseph, since you're communicating, can you see just the basic statistics slide and nothing else?
Yeah, that's all I can see.
Can you see my arrow?
Yeah, your mouse, I can see it moving.
Okay, cool. All right. Yeah, I'm just having trouble. I've got two screens going and I haven't had this difficulty. It's wanting to take over both of them with the same thing. And I want to see you guys on one screen and I want my thing to be on the other screen. And it's just not cooperating for whatever reason today.
Yeah. Yeah. It's like mirroring thing.
Yeah, so I went back and checked my display settings, but that didn't seem to clear up the issue. I think it's something with the way PowerPoint is interacting with Zoom. But anyway, seems to be working now. And so, how's everybody doing?
can you hear me now?
I can hear you now.
Okay, good. I didn't realize I had that messed up.
Hey, Traveris. Is that how you pronounce your name?
Tavares.
Tavares. Oh, Tavares.
Yes, sir.
Oh, yeah. I guess I'm getting old. I can't see that far away.
It's okay.
Just so everyone knows, you do have the ability to change your little display name up here if you want to do that.
All right, just so you know, I am having a little more of a tighter chest today than I was on Wednesday. I'm not sure why that's the case, but you might see me occasionally yawning largely. Just forgive that for me for now. Hopefully one day I will be fully healed and back to breathing like a normal human being. Dr. Judd, do you have any announcements here at the beginning?
Not for the beginning. I have announcement at the end, although for my lab students, those of you who have Thursday morning at, what time is it, 10:45, I think it is.
Mm-hmm.
You will have an email from me later today about where I want you to go, some instructions for how we're going to do that lab. So watch your email this afternoon for that.
All right, couple of things. How'd your stats homework go?
There was definitely a learning curve.
Okay, there's definitely a learning curve, sure. Did you get enough support in conquering the learning curve?
Eventually got it figured out.
Okay, so with homeworks, one of the things that came up earlier today, I want to stress this. It's in the syllabus, but in case you haven't read the syllabus with some detail, this was a preparation homework assignment. And with preparation homework assignments, what we generally do is we grade them for completeness more than correctness. The question is, did you make a good faith effort to do the assignment and get it as right as you could? Because what it really is it's to get you thinking and get you prepared for what we're going to be going through in class. So if you get some things wrong in it, it's not that big a deal. The important thing is that you push through and completed the assignment. Now, occasionally we will also give you practice homeworks. The practice homeworks are homeworks designed to allow you to practice the things that you've learned in class. And on those, the grading is basically, did you complete it, but also did you complete it correctly, because we've already taught you whatever that thing is. So if, for example, we were going to give you some practice homework on statistics, if I was to have a practice homework for statistics, I would expect you to get those things correct because you had already gotten a preparation homework assignment on it. We had then gone through it in class. And so by the end of that, you should have a much better handle on what it is that you're trying to do. Does that clarify things for you? Okay, I'm getting some thumbs up from some people on their screens. Any other feedback on the homework assignment? All right, let me ask the group a question. When should you probably start homeworks?
As soon as possible.
Okay, who was that?
Riley.
Oh, Riley. Okay, so why is that, Riley?
Because turns out it takes longer than you think.
It does take longer than you think, and this was an especially involved homework assignment. But yeah, sometimes they will take longer than you think. Sometimes they won't, but it's always a good idea. As soon as the homework is assigned, go and take a quick look at it and get some idea of how long you might think it might take, and then you can kind of schedule it a little easier. You don't have to read the whole thing, but you're like, oh, that thing is 12 pages long, and maybe I need to get started on that earlier than I was planning on doing.
Previewing is going to be a good habit to get into, whether you have to read a paper or whether you have to do homework or whatever. Before you jump into it, just stand back, look at it, see what it looks like in terms of how much effort it might take, and just get a sense of what's coming. And then once you've done that, get started.
Yeah, so I've also tried to enable this in the Moodle site. In the day stuff, I'm having it display the description on the Moodle site, so you don't have to click on it to get the description, because in that description is explanatory information that might be good to know that you don't see unless it's being displayed. So I'm trying to check that off every time I post a day's work. But today's work is in the September 2nd file. It's in a PowerPoint called Basic Statistics. And so if there are no other questions or comments, I'm going to just get started on that. All right. So just to follow up on some of the things that we talked about last time, we were talking about the scientific method last time. And I was talking to a student in my office the other day, and we were talking about what the purpose of that 2 days of talking about the Coots was. And part of the purpose of that was so that you could get this information. So you could know what the steps of the scientific method are. You would get some idea of what a mensurative versus a manipulative experiment is. You would know that this is a recursive process, such that at the end of a study, oftentimes you've come up with other questions that you want to address, and then you go through this process again, so that the process of science is an iterative process where you go through this process over and over again as you slowly learn more about the system that you're working in. But we could have just given you this rather than what some people might consider wasting 2 days talking about Coots. But in the process of going through that exploration of the Coots, what else did you gain from that? That's an open question to anybody who wants to throw in.
We didn't just learn about it, we also got to practice it and like see it work through.
Okay, good. What else?
More likely to remember it if we have to do it ourselves instead of just having to listen to it.
Okay. Anybody else?
We got like an example of how it would look in practice instead of just seeing, a list on a piece of paper.
Okay, yeah. So sometimes seeing it in action is better than just being given a list of something. Anybody else? Did you learn anything about behavior? Did you learn anything about evolution? Did you learn anything about ecology in the process of going through that? You guys are very quiet today. You guys learned nothing about any of those things?
Are you all expert waterfowl folks?
Did you learn anything about crab? Yeah, go ahead. Who is that?
Joseph. Learned about parental favoritism in Coots.
Okay, so you learned something about behavior, so about animal behavior. Did you learn anything about what?
Box and whisker plots.
I was about to say, did you learn anything about reading graphs in the process of going through that, which is an important skill in biology? So even though it was a case study that was designed to get across to you the scientific method, it also got across a lot of other things that weren't just scientific method-y sorts of things. You learned some actual factual knowledge about some organisms. You learned about one behavioral concept You learned about how people go about illustrating data. You learned, did you learn anything about the nature of a control? Raise your hand on the screen if you had heard about controls in experiments before. Okay, so what was different about the controls in this experiment?
The controls showed us different information and helped us learn about the experiment beyond just the variable groups.
Yeah, okay, this particular experiment had two controls, and the two controls were necessary to address the hypothesis in a way that wasn't necessarily the way you've maybe seen controls in other experiments that you might have been exposed to. So even though the focus was to walk you through the steps of the scientific method, you learned a lot of other stuff. Don't ignore those other things that you learned, because those things are also things that we want you to learn as we go through this. All right, so we talked about observations in the process of going through that. And I mentioned, although I didn't really talk much about kind of my distinction between trivial and non-trivial observations, those terms might seem judgmental to you, but it's just a categorization to say, you know, is this an observation that leads me down a path of further discovery and questioning, or is it a path that, an observation that doesn't? So for example, the sky is blue, the water is cool, the flower is yellow, doesn't necessarily take me anywhere that I might be interested in going as a biologist, as an ecologist. But a non-trivial observation might be something like, these yellow flowers are covered by these yellow beetles with the black spots on their back. What do you notice about the difference between that observation and the observations above?
More details.
Okay, part of it's more detail. Who was that?
Connor.
Connor, okay. More details. What else about it?
It adds like another, living thing to it.
Okay, now we're making an observation about a flower and then about another organism that's interacting with that flower. What's the benefit, somebody other than Connor, of adding that interaction into the mix?
It leads to deeper questions.
Yeah, you might ask, okay, why are the beetles there? What are they doing while they're there? Is the plant getting anything from the beetles, or are the beetles taking things from the plant? It leads you down a whole path of questions that you can ask, because you've taken a little more time and been a little more careful and a little more complete about the observations that you're making. In the same way, we can think of questions as being trivial and non-trivial, but in this case, a trivial question is a question that we already know the answer to. So those yellow beetles that are on the yellow flower is a goldenrod plant, and those yellow beetles are goldenrod beetles, and we know that they feed on the pollen, and they eat the pollen, and they eat a certain amount of the pollen. Some of that pollen gets stuck to their bodies, and so while they're eating the pollen, they also provide pollination services to the goldenrod. So we already know quite a lot about what these beetles are doing, so that many of the questions that we might ask about it, we already know the answer to. Non-trivial questions are those questions that we don't yet know the answer to, and those are the ones that we should hopefully be pursuing as scientists. We can also think about mechanistic versus evolutionary questions. It's another way of saying proximate versus ultimate questions. But a mechanistic question gets at more like, how does something happen? So when we think about hawk moths extracting, oh, I thought I changed this. Hawk moths extracting nectar from a prairie fringed orchid. Why does the hawk moth have a process that's so much longer than is necessary given the length of the nectary? So, sorry, excuse me.
God bless you.
Yeah. That still hurts. So here's a prairie fringed orchid, and this green thing on the back is the nectary. It's a place in the plant that produces nectar, and presumably, why would a plant produce nectar?
In order for bugs to pollinate.
Yeah, so it's an attraction to get a moth to come to the plant, extract the nectar, and while the nectar is being extracted, the moth presumably gets some pollen on its body and then carries that pollen to the next prairie fringed orchid. So one of the questions would be, how does it do this? Well, it does this with a proboscis. The proboscis is long. Well, why is the proboscis long? Well, the proboscis is long so that it can get to the nectar that is at the bottom of this nectary. But what do you notice about the proboscis of these two moths?
One seems to be like full grown and the other seems to be young.
Okay, so when insects reach this stage, they're adults. They're not going to grow any more than they currently are. So this is actually two different species of hawk moths. One is big and has a really long proboscis. One is smaller and has a shorter proboscis. What was the question I asked?
You asked about the difference between these two moths. I think you kind of.
Oh, I kind of answered my question. So one of the differences between these two moths is they're different sizes, not because they're adults and juvenile, but because they're two different species. But this one has a really long proboscis and a really short proboscis. So both of these species get nectar from a plant using their proboscis, but a deeper question, an evolutionary question might be, why does this species have a really long proboscis and this species have a really short proboscis? Notice that this proboscis is about the length of the nectary. This proboscis is way longer than the nectary. It doesn't need a proboscis that long to access the nectar from this plant. Why might you want a big proboscis like that? Would be one of these evolutionary questions. So anybody have any ideas why you might want a long proboscis?
Maybe so it doesn't have to land on it or be close to it to get the nectar.
And why would you want to avoid touching the flower to get the nectar?
To spread the pollen.
Yeah, the pollen weighs something, and so however much pollen you have on it, that's additional weight. And so if you're carrying more pollen, you might be expending more energy in flying. And so this might have evolved as a way to steal nectar from the prairie orchid without having to pick up any pollen from the orchid. And people have studied this actually. This guy at the North Dakota State University has studied this and found that that's actually precisely what these guys are doing. And so why the evolution of the long proboscis? Well, it's to get the nectar without incurring the cost of carrying the pollen. So evolutionary questions go beyond these sort of mechanistic questions. Everybody kind of clear on that distinction? All right. So ultimately, your questions should be based on observations that you make. They should be grounded in what is already known about the biology of that system. So sometimes refining questions requires going and doing book research or reading primary literature articles about that system. They should be linked to an underlying conceptual framework that informs the question and points to potential answers. The questions that we just had was about a particular interspecific interaction having to do with pollination. And so you would want to be reading information and papers about pollination ecology if you had questions about that system. It should lead to a set of, your questions should be capable of leading to a set of alternative hypotheses that are mutually exclusive of one another. And hopefully these questions would also point you towards a set of experiments or observations that would allow you to answer the question. If your question isn't doing all of those things simultaneously, chances are you haven't come up with a very good question yet, and you need to spend more time formulating a question. And as Trinity from the Matrix says, it's ultimately the question that drives us. So the better question you develop as a scientist, the better experiments you're going to do, the better answers you're going to come up with, and the more productive you're ultimately going to be. Of course, in our case, the answer is not the matrix, it's something biological. So questions about the whole COOT thing, the experimental design, asking questions, anything about that stuff before we move into statistics?
I had a question, not about this specifically, but I was wondering, is this PowerPoint available to us?
It's on the Moodle site in today's folder. Yep.
Okay.
It should always be there. Yep. Any other questions? All right. So in terms of statistics, my goal for today is to help you understand the importance of the central limit theorem and the empirical rule in statistics, and why are these two things so important. And the reason is because it helps us relate information that we gain from samples to populations. And hopefully the homework helped you build the groundwork for getting that idea. I want you to understand how to calculate a 95% confidence interval, because 95% confidence intervals are important. So I also want you to understand why 95% confidence intervals of the mean are preferable to other measures of dispersion like variance or standard deviation. And if you haven't already gotten an inkling of that from the homework, hopefully you'll get more of that today. Understand how sample size affects the width of the 95% confidence interval and one's ability to make inferences and decisions about the data that you have. And by the end of this, you should all be able to accurately estimate whether two means might be different given their means and their 95% confidence intervals. And hopefully you're well along the path of being able to calculate those things effectively on your own. And the important point about the confidence interval is a lot of times when people think about confidence intervals, they think about the overlap of the interval itself rather than thinking about the inclusion of means in confidence intervals. And I want to drive home the point that it's the inclusion of the means that matters, not the overlap of the ranges that matters. And this will become clearer at the very end. So that's the last thing that we'll talk about. So one of the main reasons that we have to do statistics is because everything out there in the world is variable. This is especially true of biologists. And so if you go out and study anything in a natural system, whether that's a natural system out in nature or it's a natural system that you've brought into the lab, it's going to have variation associated with it. Variation can be due to environment or genetics, or variation in data can also be due to human errors. But often it's the variation in environment and genetics and things like this that is of interest to biologists. So sometimes people think of variation as a bad thing, but in many cases, this variation is actually really important to biologists. We've asked you in the process of going through and making observations to think about variation among individuals, because that's one of the things that's going to be important in the lab, but it's also important in all areas of biology. So the question is really, how do we deal with variation? And that's what statistics has a lot to do with. So oftentimes we use the term error to refer to this variation, but there are two kinds of error. There's sampling error, which is simply the deviation of a sample mean from a population mean due to the fact that you didn't sample the entire population. If we really wanted to know what the average value of some variable was, we could actually go and measure the entire population. But oftentimes we don't have the time or the resources or the patience for that matter to go and measure the entire population. So we go and we grab a sample of the population and we hope that that sample tells us something about the population that we grabbed that sample from. That's a totally appropriate kind of error. It's called sampling error, and statistics helps us deal with that error. But there's another type of error that we want to also be able to deal with, but we want to deal with before we get to the point of gathering data. And this is measurement error. Measurement error is when you gather data in ways that make your data inaccurate. So we were measuring a proboscis on a moth that was, 5.4 centimeters long, but because there was something wrong with our measuring instrument, it isn't actually 5.4 centimeters long, it's 6.7 centimeters long. So we measured something and we got the wrong answer because we messed up the measurement in some way. We were using an inaccurate measurement, we were using a non-standard way of doing it, whatever. Those kinds of things you can fix by standardizing methods, by making sure that the instruments that you're using are calibrated and things like this. So I've used this term inaccurate. So I've referred to the accuracy of one's measures. And one of the things that we see every year, so your guys are going to do this, but realize every entering class of students has done this before you. We oftentimes see students misusing the term accuracy. So they'll say in their final presentations, oh, we could have taken more data to make our measurements more accurate. Taking more data doesn't make your measurements more accurate. What taking more data does is it reduces the size of your 95% confidence interval, but it doesn't do anything to the accuracy of those data. So I want you to take just a minute and write down on your paper or on your iPad, what would your definition of the terms precision, accuracy, and repeatability? What do those terms mean to you? I realize this is a really uncomfortable silence, but I'm looking at people's faces to get a gauge of when you're getting finished. So just continue working. Okay, that's been about a minute per definition. Who wants to share their definition of precision?
I said that if something is precise, the results were similar to each other.
Okay, repeat that again.
If your results are precise, the results are similar to one another.
Okay, does anybody want to comment on that definition? Do you all agree with that definition? Who's that?
I thought her definition would be more useful for accuracy rather than precision.
I actually don't think it's useful for accuracy or precision. What would it be
Really?
most useful for?
Oh, repeatability, probably.
Repeatability, getting the same answer over and over again. Now, if you have an incorrect instrument, you could measure the same thing a similar thing over and over again, and it would give you the same answer, but because your instrument was not calibrated correctly, it would just be giving you the same answer over and over again if it was very repeatable.
I got
So
this really nice ruler from the Missouri Department of Natural Resources. And as you can see, the 20 to 30 millimeter range is not 10 millimeters wide. So here's an example of an improperly calibrated ruler where there's 7 millimeters between 20 and 25. And
Wow,
it wasn't
that's
until I started measuring things that I noticed it.
Well, that's kind of messed up.
Yeah.
Yeah. So you could get a highly repeatable measure from that, but it wouldn't be accurate. So Michaela, I think I would put your definition under repeatability rather than precision. So who wants to take a shot at precision then?
I said it's the ability to orchestrate or perform a task fluently.
Who was that? Dominic?
Yeah.
So repeat that again.
Ability to orchestrate or perform a task fluently.
That would maybe precision in terms of like figure skating, but I'm thinking of performance in terms of measuring a variable. And precision is very important in figure skating, but I don't think we're going to be doing a lot of figure skating in this class. Somebody else take a shot at precision.
Maybe like a grouping of your results.
A grouping, so like a tight grouping of your results? What does that sound more like?
Yes.
Getting the same results over and over again. That's repeatability again. Yeah.
For precision, I'll put data that is like steadily close or on target through duration.
Okay, so if the data that you're gathering are on target, and I assuming that you mean by on target, you're basically saying that it's giving you the correct answer, what would that be? Is that precision or is that something else?
Accuracy.
That's more like accuracy. Who was that?
It's Connor.
Connor. Okay, cool.
Yeah, for precision, I put like, the values aren't necessarily correct, but all of the measurements you are taking are kind of close to each other. That's what I had for precision.
That would once again be repeatability in my mind. So clearly, this is not something that's easy to wrap our heads around. So have you had conversations in other classes in your career, in high school or whatever, about these terms? Yep, some of you. Did the person who taught it to you use an analogy using archery?
Yep.
Oh yeah. You know how I know this? Because when you search for analogies for precision, accuracy, and repeatability on the web, everything has to do with archery. And here's my problem with the archery analogy that people use. So here's just one of like 5,000 ones I could have pulled off the web. This is showing low accuracy and low precision. And I would argue that this is actually just low accuracy and low repeatability. This one is low accuracy, and they say it's high precision, but it's not really any more precise than this one is because each of those darts has a little pointy tip on it. This is low accuracy and high repeatability because every dart is going to roughly the same place. This is higher accuracy, but then once again, it says it's low precision, but really it's just more accurate and somewhat more repeatable than this one over here was. And this says high accuracy and high precision, but it's highly accurate, but it's also the highest repeatability of any of these panels. Why is precision not really illustrated in this figure?
Because precision is more about the narrowness, or like how close you can get to an exact number instead of like a widespread on that, I guess, quote unquote target. Like you said.
So precision is all about how finely divided the measurement scale is that your instrument can measure. So I'm going to give you another analogy, a different analogy. It's kind of related to this, but it involves firearms, because we might as well be playing with firearms if we're going to be doing anything. So here is my accuracy and precision. So starting on the far left here, I'm shooting this thing with like a .44 caliber. I'm making big holes in the target. So I'm not very precise. I'm not very accurate because I'm not hitting the bullseye, and I'm also not very repeatable because these gunshots are all over the target. So this includes both precision and accuracy and repeatability, and the far left one is none of those. It's not accurate, it's not precise, it's not repeatable. The second one here has the same level of inaccuracy. I've put these gunshots in all the same places. It's not terribly repeatable because they're not clustered together, but what it is it's more precise, because now rather than shooting this target with a .44 caliber, I'm shooting it with a .38. Smaller bullet, more precise. I'm hitting a smaller portion of the target. The third one is now a .22 long rifle, but I'm not very accurate with the long rifle, and I'm not very repeatable because these things are not in a nice tight cluster. The fourth one is not very accurate because I'm not hitting the bullseye, but it's more precise than the first one because the size of each of the gunshots is smaller, and it's certainly more repeatable than any of the others because they're all nice and tightly clustered. The last one here is the most accurate because every one of them is hitting the bullseye. It's arriving at the correct place. I'm shooting at that thing with like a BB gun or a pellet gun. So the precision is very fine. And it's also very repeatable because they're all clustered together in one spot. Hopefully this is a better analogy than the previous analogy because we can use calibers of bullets to talk about precision. Ultimately, when we're gathering data, we want to be as precise as we need to be. And you don't always have to be super precise. You can be as precise as is necessary for a particular question. But you always want to be accurate, and you always want to be repeatable. If I measure a variable and Dr. Judd measures a variable, we want those variables that we measure to be comparable to one another. And the way we do that is by making methods that allow us to both be gathering the data in the same way so that his measures would be the same as my measures if we were measuring the same thing. Does this make sense to you? Nod yes or shake no. Okay, all right. So this is an example of how we sometimes sacrifice precision to get accuracy and repeatability. So this is a thing called a Daubenmeier frame. We use it in plant ecology to measure percent cover of plants. And it basically breaks this frame up into 25% brackets and then a 5% bracket down here. So we can put this over a community of plants out in the field, and we can very quickly and easily place these plants into a set of ranges. Is it 0 to 5% cover, 5 to 25% cover, 25 to 50% cover, etc. And so if you go out into a field and put this over a bunch of plants, you can kind of look at frame number one here and say, okay, this spiky plant that's coming up from a common root, you could put 1, 2, 3, this half of this one and this half of this one. They would fit. They would not fit inside of this first quarter, but they would all fit inside of this second quarter. So they're in the 25 to 50% category. The nice thing about the Daubenmeier frame is it's easy because you're just putting them into categories, kind of mentally grouping them and saying, well, how many of them would fit in which section of the frame? It's highly repeatable because if I ran a Daubenmeier frame and Joseph ran a Daubenmeier frame, we would come up with the same answers. But what it isn't is it's not particularly precise because we don't have the scale divided up very finely. It's in these rough, large categories. So we've sacrificed precision in favor of speed, accuracy, and repeatability. And that's a perfectly legitimate sort of trade-off to make in measuring things. Dr. Judd, you look like you wanted to say something.
No, you're there.
Okay, all right, cool. So one of the points of today is to help you get a basic understanding of statistics. So I gave you this card drawing task, and none of you came by to borrow my cards, because if you had, you could have used the reptile and amphibian card deck, but you found cards on your own or you went up to lab. And one of the first questions on the homework was, when we look at a deck of cards, this is a full deck of cards from ace to king. What kind of a distribution is this? Is it a normal distribution? Is it a binomial distribution? Is it a random? No, not a random distribution. A log normal distribution, a uniform distribution. What would this be?
Uniform.
Yeah, who was that? Tessa? Okay. It is a uniform distribution. Tessa, tell us why it's a uniform.
Because it goes up in even increments.
Okay, explain that a little more.
So there's a clear pattern of like it increasing.
That's actually not what we would mean by a uniform distribution. Somebody take another shot at that.
There's like different sets of the exact same amount of cards.
Yeah, so there are always 4 aces. There's 4 2s, there's 4 3s, there's 4 4s. There's the same number of each of the individual numbers, so that it doesn't take a bell-shaped form. It's just like a flat plateau where all of the things in the data set, in the population, are represented equally. That's what a uniform distribution is about. And then I asked you to start drawing cards from this uniform distribution, and I asked you to do it in a particular order. And so I asked you to first draw 10 cards, or 2 cards from 10 samples. And when you do a histogram of this, your histogram turned out differently than mine, but they're probably all similar. Does this look like a normal distribution of sample means from this? So I asked you to draw 2 cards out, take the mean of those 2 cards, and then plot it for 10 samples. Is this a normal distribution?
Yes.
It doesn't look that normal to me, because the differences between them are just 1 to 2 in terms of their frequency. And there's a peak. It doesn't really center on the average, on the population mean of 7. And it's less normal than I would like it to be, mainly because it's only 10 samples. What does the central limit theorem tell us from your homework assignment?
You need a large number of samples.
Okay, so, but back up, what does it tell us in theory? It tells us if we go to a population and draw out a bunch of samples, what will those sample means ultimately give us?
It like tells you about the population as a whole.
Okay, so it will basically give us a distribution of sample means. Where will that distribution of sample means be centered? Is this Michaela? Who is this talking? Audrey. Who was talking a minute ago?
It's Katie.
Oh, Katie. Okay, Katie. So if you get this distribution of sample means, where is it going to be centered over?
I don't know.
Somebody help Katie out.
It would be centered on the population mean.
It should be centered on the population mean. Is that Elena?
Yeah, it was.
Okay, cool. I'm still trying to learn your names. It's just harder when we're online. So it'll be centered on the population mean, right? And it will be what shape?
It should look like a bell curve.
It'll look like a bell curve. It'll look like a normal curve. So this isn't centered on the population mean, because the average of this set of 10 samples for my data set was 6.8. So it's close, but it's not exact. And it certainly doesn't look like a normal curve. So what happens as we increase the number of samples? Well, as we increase the number of samples, because we have more samples, we might end up getting closer to the population mean. Although you can see here that we're still off the population mean. Does this look like a terribly normal shaped distribution? No, it looks kind of wonky. It's got a couple of big peaks, but then everything else is just kind of low frequency because we're still dealing with only 2 cards and we're only upped our samples to 50. So we only have 50 sample means that are illustrated here. But we can increase our sample size so that we're drawing 5 cards out of the population of the deck of cards. And the distribution of the data begins to start looking a little more normal, but you see that the distribution of sample means, its average is still like 0.2 away from 7 if you look at the x-axis. Well, we can go back to 2 cards, which is still a small sample size from the big population of a deck of cards, but because we're now drawing 1,000 samples, this was the computer simulation that you did, we see that this begins to kind of look like a normal distribution, doesn't it? But it's not because we've increased our sample size, it's because we've increased the number of samples that we've drawn from that population. So this is the distribution of sample means. We see that the average value of that distribution is still not at 7 because 2 cards doesn't give us a very complete view of what the population is because we're taking really small views of the overall population. You'll also notice that the variation in this distribution is really wide. Once again, that's because we're only drawing 2 cards out of the population. What's going to happen if we increase it to 5 cards and 1,000 samples?
Would the graph narrow?
What's that?
If we increase the cards to 5, would the graph narrow up?
It'll narrow, and what will happen to the correspondence with the population mean?
It would get closer to 7.
Should get closer to 7. So let's see if that happens. So now we have 5 cards and 1,000 samples, and we see that the average has crept closer to 7, and the overall variation of the distribution of sample means has shrunk in. It was way out here from just below 2 to about 12, and now it ranges in terms of 2 standard deviations of the distribution of sample means from like 3.7 up to 10.1. And so the population is getting narrower. The distribution of sample means is getting narrower. What happens if we increase sample size to 10? 10 cards in 1,000. Somebody other than Cody.
Narrow, get even more narrow.
Oh, who's that?
Connor.
Connor. Okay, it should get even more narrow. Now the average of the distribution of sample means is right at 7, and this range of means generated from the population of the deck of cards has gotten narrower. What's going to happen at 20?
More narrower.
Yeah, same song, 7th verse. It's going to get narrower, and when we go up to 30, now when we're taking a sample of 30 from a population of 52 cards, we have a really good view of what the cards are like in that deck because we've measured now a significant proportion of that deck. So we know pretty well what the population mean is, and the amount of uncertainty that we have around that estimate of the population mean is really small because we now have this really big sample size. So when we take a single sample out of a population, we can actually use this relationship, the central limit theorem and the empirical rule. The empirical rule tells us what percentage of the sample means will be within 1 standard deviation, 1.96 standard deviations of the mean. We can use those 2 things to use the sample mean to allow us to build a range of values that we can expect to find 95% of all sample means you could draw out of that single population. So the confidence interval, the 95% confidence interval of the mean, basically says what's the range of values in which we would expect to find 95% of the sample means. So when we're asking questions about our data, we're not asking our questions about the population. We're asking about the distribution of sample means that came from that population. And that's an important distinction to think about. Because our population, what's the shape of our population? Is it normally distributed like this? Tessa?
Sorry. Yeah, so as the population sample increases, then so should the accuracy around the mean. So it should get narrower, right?
Yeah, so that's what I'm illustrating, but the population that these samples came from, is the population normally distributed? What's the population distribution?
It's uniform.
It's uniform. Thank you, Cameron. Tessa had the answer earlier, which is why I went back and asked her. So this is a uniform distribution. So the empirical rule tells us at 1 standard deviation, about 68% of a normal curve is in there. And at 1.96 standard deviations, 95% of a normal distribution is in there. So the empirical rule doesn't tell us anything about this uniform distribution. But because we know that the central limit theorem tells us if we sample this uniform distribution, it's going to give us a normal distribution of sample means, we can then use the empirical rule to say, well, about 2 standard deviations out from that, we can expect that to encompass 95% of the distribution of sample means. So we need both of these things to allow us to do statistics. The central limit theorem tells us that the distribution of sample means will be normally distributed and then the empirical rule tells us things about the normal distribution that are useful to us. And what it tells us is that 68% of the observations, 68% of the distribution of sample means lies between plus or minus 1 standard deviation of the distribution, and 95% is plus or minus 1.96 standard deviations of the distribution. And so because we know that about a normal distribution, we can make inferences about how many sample means will be within a certain range of numbers. But that only helps us if the central limit theorem gives us a normal distribution to work with to begin with. And so that's the reason that these two things are super important.
Remember, those confidence interval calculations are going to, the confidence you have that your means are going to be within a standard deviation require that you take the same sample size. You take so that your mean is your sample mean is derived from 30 cards every time in the case of the slide on your screen here. And that should be intuitive, right? If you pick up one card and it's a face, and you're only going to pick up one more, 2, it doesn't necessarily have to be an ace. It could be another face card, then you'll be way out on the tail. But if you're picking 30 cards and you get a face, odds are one of the other 29 cards you draw is going to be an ace or a 2. So that's going to average out, and you're going to be closer to this mean.
Yeah. And so Tessa was correct in that the larger the sample size you have, the clearer picture you have of what this distribution of sample means is. And as a result, you have much more confidence in where the mean of that distribution is, which is why this distribution sucks in on itself over time. Okay? Questions about this idea?
Okay, I have a question.
Yeah, go ahead. Yeah, Tessa.
will there be more information about like this type of statistics in our textbook that we can look at? Or is there any way that we can get some more practice?
Unfortunately, the textbook is not very strong on statistics, but Nathan is about to look in there.
Yeah,
They do talk about this some at the beginning of the book. We had box reading in the readings, but it doesn't go into nearly this level of detail. So you're saying you would like some practice with this?
Yeah, it's just I'm having a hard time with the vocabulary, just keeping up on the definitions. So I was wondering if there was a place that I could like.
Okay, so part of this is defined in the homework. So in the process of telling you what to do as you work through the homework, definitions for these things were kind of embedded in there. So go back and check that. Let me also try and put together sort of a working glossary for this, so you kind of know you have a central repository for referencing. Would that help?
Yeah, that would be.
I'll just add in here, I was talking to one of you earlier about this, and that's a good instinct. Getting to know the vocabulary around these equations is going to be much more important and valuable than trying to memorize equations.
Yeah, so Nathan is getting to a point that I'm going to make here in a few minutes. The act of working through how to do it is much more useful than memorizing the equation. I know what the equation is now because over the years I've kind of internalized the process of calculating a mean, calculating a variance, and things like this. So yeah, Nathan's right on point with that. So at the end of the homework, I gave you a little tiny data set, and I asked you to calculate the mean, the variance, the standard deviation, the standard error, mean, variance, standard deviation, standard error, and the 95% confidence intervals. So I just took those data and I started plotting them. So when we look at the beak lengths of finches from Academy Bay, because we only have a sample of 5, the histogram, the frequency histogram, doesn't really look like much because there just aren't that many observations. But notice on the x-axis, it goes from like 9.75 to 10.75. One of the things when you're reading a graph you should always look at first is what's on the x-axis and what's on the y-axis, and what are the numbers on those axes. Because when we go and look at El Garapatero, we see that it also doesn't look like a normal curve because these are just the raw data. But it starts at 11.25 and goes up to 12.25. But once again, there's just only 5 observations here. Well, we can take these observations and we can plot them on the same graph. And what we see is that it looks like the distribution of birds in Academy Bay doesn't overlap with the distribution of beak lengths in El Garapatero at all, which might make you think that those birds are fairly distinctive in terms of how long their beaks are. Well, we could actually take those numbers, those measurements, and calculate the mean, and calculate the 95% confidence interval of the mean. And when we do that, we can make a graph that looks like this. This is what I was hoping you would do for your last figure. If you didn't get this for your last figure, it's not that big of a deal. Some of you did it in bar graphs and things like this. That's fine. But what we see is that Academy Bay has an average beak length of a little below 10.5. El Garapatero has one that's like 11.7 or something like that. But what you see is this is the 95% confidence interval of the mean for El Garapatero, and this is the 95% confidence interval for the mean of Academy Bay. So when you calculate these 95% confidence intervals, what it basically says is if I go back to Academy Bay and I take 100 samples of bird beaks, 5 bird beaks each sample, and calculate the means of all of those, 95 out of 100 of those means should fall within this range. If I go to El Garapatero, I gather up 5 bird beaks, for 5 birds, I measure their beaks. I take the average of that. I do that 99 more times. 95 of those sample means should fall within this range. 5% of them will be outside of that range. That's what the 95% confidence interval of the mean says. If I take 100 samples from a population of size 5, in this case, because we have 5 finches, 95% of them will fall within that range of values that is the 95% confidence interval of the mean. Now, the reason that I want that to be the situation, and that's what the central limit theorem and the empirical rule tell us. The reason I want that to be the situation is because I actually probably am not all that interested in what Academy Bay beak finches are like. I'm probably not all that interested in what El Garrapatero beak finches are like. What am I probably most interested in?
Maybe the difference between them.
I'm interested in, are Academy Bay finches different from El Garrapatero finches? Well, because I know that 95% of the sample means from Academy Bay would fall within this range, is it very likely that this mean came from Academy Bay? This El Garrapatero mean came from Academy Bay?
Yes.
But it's way outside the 95% confidence interval. If this is where 95% of the sample means from El Garrapatero would come from, is it likely that this mean from Academy Bay came from that population of El Garrapatero birds? No, because it's way out here, way far away from that range of values. So at the beginning, when I was going through the learning goals, I said that making decisions about whether or not two things are different has to do not with whether or not these ranges overlap, it has to do with whether or not this mean falls within or is included within this confidence interval, and whether this mean is included within this confidence interval. So it's a matter of, are the means included in the confidence intervals of the other populations, or are they excluded from it?
If you're writing this in your notes, I suggest that you sketch this where there's no overlap at all between the two lines. Sketch one where there's some overlap between the two lines, but the means are not captured within the other set. And then draw one where there's total overlap and both means are overlapping. And then highlight these two, show a difference, and this one doesn't.
So right now, this mean is not included in this confidence interval. This mean is not included in this confidence interval. If the data were more variable than they were, let's say that this thing is really variable. El Garrapatero is super variable. We don't have a very good idea of where the mean is, which is why the confidence interval is so high. And if Academy Bay is really variable, and now I can't even do it because I run off the screen, we don't have a very good idea of where the mean is. In this case, these things would not be considered different because this mean is now included in the confidence interval of this mean, and this mean is now included in the confidence interval of this mean. So we would say that we have so little information about these things, we don't have very precise, we don't have very precise measures of where the actual population mean is, and so our confidence intervals are really big and they encompass a lot of different values. Now, why do we have such not great, well, so in this case, we would conclude that these beaks are different between these two populations. And now I need to learn how to erase erase. So it would show that these two things are different. But what's the problem with only having 5 finches?
It's not really enough data to use.
It's not really enough data to get a clear view. So I actually randomly selected these 5 values out of a larger data set. And because I think Dr. Judd has a couple of announcements for you, we'll start there on Friday if Dr. Judd can give me about 5 minutes to finish up.
Yeah, that'll be fine.
Okay, so we'll end this here today, and Dr. Judd has some announcements.
All right, so you're going to see a few different things show up on Moodle for our activity on Friday. But for right now, the only thing I want you to access is the text file that's in the folder for today. So your assignment for today, your homework for Friday, which is your assignment today, is to download that text file. And what I want you to do is find a word cloud generator online. It's pretty easy to do. You just Google word cloud generator. You'll find some options. Get yourself a free one. There's several out there. And then you're going to either copy the text in, copy paste it, or it might ask you to upload the text file. So you'll click choose the file from, and navigate to where you put it in your downloads folder or whatever, and then it'll upload that. There's one of them is called Wordle. There's another one that's a website by Daniel Frazier that's pretty good. There's several out there. You'll find choices. You don't have to have the same one as the other folks in your group. But you don't have to read the text. I just want you to download it and make a word cloud. I'm also going to put in a paper and then the slideshows, but I don't want you to look at the paper until we're in class when we're going to read it.
This is another one of those places where we're controlling the flow of information, because to get access to the information beforehand kind of wrecks the activity that we have planned for you. Also, you should know I'm keeping track of who's contributing on my rubric here. And so I'm also assigning points to people. I haven't done it for today, but I've been keeping track of people for today. So on Friday, I'll also update you on kind of where different groups are standing. So keep participating and know that we are keeping track of it. All right. If you have individual questions about what we went through today, email me. At the end of what we do at the beginning of class on Friday, I'm going to ask you some questions to make sure that you're kind of tracking with the information. And we'll see you on Friday. Hey, Nathan, I realize you're going to class. Let me kill this first.
I actually, I'm not sure if I will be able to attend class on Friday because I have a family issue.
Okay.
So in the event that that happens, I might be able to make it, I might not. Should I just watch the screencast?
Yeah, sure. Yep,
okay, and
exactly.
then take notes on it? Okay.
Yep.
Thank you.
And talk to your friends and whatnot. Sure.
Okay, perfect. Thank you.